This brings me to the crux of what I wanted to talk to you about. I've been wanting to talk to you about for many years now, and that is when we were going through all of the COVID madness, I felt like I was in the twilight zone because I've got colleagues, I've got friends who are molecular biologists.
You and Byron Bridle both were in the what the hell?
Yeah, we, as I said on the onset, molecular biologists are intimately familiar with this technology that you just described. We use it day in and day out, and all of a sudden we've got talk of mass production, huge scaling up of this technology to deliver mRNA as a means of producing an antigen.
Your fellow Canadian, Dr. Peter Cullis, gets the gold star for advancing this. And I knew Peter from back in the liposome days. Let me just give you another story. So I knew Peter from back in the day. And as this was breaking, he would still talk to me. He won't talk to me anymore. And I called, I don't know why, tongue in cheek, but I called him up. I'm sure Peter is got a very nice yacht or whatever now. He's got all the money in the world, and God bless him. He had been working in liposome technology development delivery for decades. And I called him up early on. I said, Peter, what's the nature of these formulations? And what is it that is making them specific, and why are they working in vivo? Because I could never get them to work efficiently in vivo. What they mostly did was trigger neutrophil influx. They're insanely inflammatory. And he said, well, we have a new molecule that we've engineered that is the cationic lipid component. It's a branch chain, much more elaborate than what was used back in the day, which are, dual chains, hydrocarbon chains. And it has a tertiary mean rather than a quaternary mean. Remember, I told you this was going to come back. Yeah. So the tertiary mean is very much pH dependent about whether or not it's going to be charged or not. And so it turned out that tertiary means, he believes, were essential to in vivo activity, because one of the problems when you use this stuff in vivo is that it interacts with extracellular proteins and other things in the extracellular milieu, and it can inactivate the ability, this key property of needing to have a net positive charge to drive it to association with cell membranes. So he said, well, we tried, a zillion different cationic lipids, and that's true. I mean, the whole field did. I did in my laboratory.
We experimented with some ourselves as well.
Yeah. So he came up with a new cationic lipid structure. It was a tertiary mean. And the other breakthrough was that he formulated it with diallyl phosphatidylethanol. I'm sorry. Yeah, no, not dope. I'm sorry. With I'm blanking suddenly. The molecule that's used in car radiators.
Glycerol.
No. It's not glycerol. Oh God, I'm sorry. I'm blanking on this.
It'll come to you.
It's a short side chain hydrocarbon linked to a oh God damn it, can't remember the name of the molecule. It's a hydrophobic. Well, it's a hydrophilic molecule that is used in stealth liposomes. And it gets highly hydrated, and it'll come to me probably 10 minutes after we finish our broadcast. It's highly hydrated, and what it does is it creates, remember I was talking about structured water?
The gel.
So it creates a structured water shell around the lipid nanoparticle. And what he did, that's fundamental to stealth liposomes, which when they're hydrated like this, they will pass through the liver. And then they do something called passive targeting, where they will accumulate in leaky capillary beds. Leaky capillary beds being a key characteristic of inflammation and cancer. So that's stealth liposome technology. And he used a version of stealth liposome technology that instead of a longer anchor lipid that locks those molecules in. He used a short chain hydrocarbon that made it so that when you formulated the particles, they would not coalesce, which is a problem with cationic. You know this in the laboratory. You kind of often have to make up the cocktail and then use it right away, because otherwise it'll aggregate and flocculate. Yes. And, it'll turn into large aggregates and they don't work anymore. And so his breakthrough was in particular, to use these short chain hydrocarbon linkers so that in the vial, the products don't aggregate. And upon injection, that tends to falls off because it's it doesn't have as much of the hydrophobic bonding characteristics. And so it tends to fall off, and you end up with a period where you have an active particle that will still be driven to associate with cell membranes. And his assertion was that these advances resulted polyethylene glycol. Finally,
of course,
in my brain. So these are polyethylene glycol linked to a short chain hydrocarbon that are used in the formulation process in such a way that they remain bound in the vial and then fall off at some point in time after injection, which is why if you have a polyethylene glycol sensitivity, you can get an allergic reaction to these things. That's one of the adverse events. And like when I got my second dose of Moderna, which was the one that triggered my adverse events, I got one of the bad batches. I could feel shortly after the injection a tingling in my fingers. That's this release and distribution of this cofactor molecule. But it's what Peter in his group came up with to kind of make this whole engine run. And then Peter asserted to me at the time that this is what enabled them to solve the distribution problem, which is another one of your questions. Didn't they know that this would go all over the body? And he had actively promoted in manuscripts, published manuscripts, that this combination had resulted in a particle that would only would remain within at the site of injection and in draining lymph nodes. Presumably, macrophage would acquire the particles at the site of injection, pull them into draining lymph nodes. In the case of humans, that would be your axillary nodes. This is based on a small number of mouse experiments. That was not supported by the actual data that Pfizer and Moderna generated early on in their preclinical packages that showed this stuff would go all over the place, which is a whole nother story. They used the least sensitive method for detecting their reporter gene, luciferase, which was whole body imaging. It turns out that you can administer these preparations. I did it, for years. I was kind of one of the pioneers in this area. You can administer your gene delivery preparation encoding luciferase into your favorite rodent, and then which has ATP necessary for the reaction. And then you can inject the substrate for luciferase, which is called luciferin, extracted from fireflies largely. And you'll have a light, you'll produce photons. And you can take your mouse and put it in a lightproof box, with a highly sensitive photomultiplier tube camera called a VIM camera. That's used like for satellite technology, spy cameras and stuff. And you'll detect the photons that are coming out through the mouse and hitting the camera lens. And you can get an image of a mouse that is glowing in various places. And you can appreciate this is not a very sensitive technology compared to like dissecting the mouse and lysing the organs, but that's not what they did. In order to detect the distribution and fool the FDA, what they did was they used a VIM camera, a light box set up, and they looked at whole body imaging. And then as Byron Bridle, your colleague there at Guelph, documented, not only did they use the least sensitive technology for looking at the biodistribution of the gene expression in the products, but then they edited the images to cut out the inconvenient parts. And that's what they submitted to the FDA. That's kind of where all this led. So I'm sorry, Peter. It's now, as you know, documented. You were wrong. The formulation that you came up with all of its masterpiece of engineering and insight, did not result in a product that remained localized to the site of injection and the draining lymph nodes. It goes all over the body. And perhaps the hydrocarbon side chains need to be shorter. I don't know. I think that there are fundamentals to the tech that are going to be difficult or impossible to overcome. And probably, if you're going to ask me the question that I get asked from time to time, can this ever be made safe? My response is something along the lines of, well, pigs could fly if they only had wings, and who knows what's in the future. One of my former postdocs that came from the Karolinska, named Peter Berglund, has continued working in this area. And Peter asserts that he and the group that he's working with now have a new delivery technology that is safe and that bypasses all these problems. And so I don't know what the future holds, but we're not going to get around these fundamental issues of pseudouridine, frame shifting, the potential interference, and other fundamentals that reside in the nucleic acid part of this. And we may get around the toxicities and problems associated with the lipid nanoplex.
I'd like for you to, if you don't mind, to comment on two things that occurred to me all those years ago as I was watching Nobody Say Anything in terms of my colleagues. And one of the obvious ones is when you're working with DNA or RNA, is the variability of the product that you're making, and then the exaggeration of that variability when you start to scale it up even larger, right? It's one thing to do this on a benchtop, working with cells, running your research, but when it's time to scale this up in bioreactors, hundreds of thousands of liters at a time, I'd like to hear your comment on the issues there.
So, and this gets where the rubber hits the road is what's referred to as process 1 versus process 2, among other things. So the initial clinical trials, I think it's of the BioNTech Pfizer product, employed a manufacturing process in which the templates were produced using PCR. And that has its own inherent flaws in terms of integrity and so purity and identity of the template. But it turns out that's expensive and not very scalable.
Right.
And so although they ran the initial clinical trials using a PCR-based template to produce the RNA, as Kevin, I think, again, I'm going to mention, he's not a doctor, Kevin McKernan, who has his own company because he was at the core of a lot of the technological innovations that enabled the Human Genome Project, and he made a lot of money. And now he just gets to play and do whatever he wants to do. And among the things that he wanted to do was find out whether or not there are contaminants in these, quote, RNA preparations. And so he and some others, but I think Kevin gets the gold star as far as I'm concerned for tip of the spear, started sequencing the residual DNA present in the vials. And then as an experienced genome person, he ran some standardized software to see if he could reassemble those fragments because they're random after he'd sequenced them. And lo and behold, what he found was that what had happened was that they had started using bacterial plasmid DNA and for the production of the RNA. So they had gone back to using essentially identical tech to what I had used back in the day. and the same enzymes, same basic structure.
Benchtop stuff.
Except, notable exception, that they used what was called back then a shuttle vector. And these are bacterial plasmids that not only have now we're going to talk insider molecular biology wonky stuff. They not only had an origin of replication for the bacterial vector in bacteria, but they had an origin of replication from mammalian cells so that it would reproduce. These circular DNAs would be able to be amplified and manufactured once they were inside a mammalian cell. So that's a shuttle vector because it can go from bacteria to mammal cell and replicate itself in both environments because it had the switch to replicate in bacteria and the switch to replicate in mammalian cells. What was that switch? It was the simian virus 40 origin of replication.
Much talked about.
In addition to the SV40 origin of replication, it also included a promoter enhancer cassette associated with that SV40 ORI. It is not the entire SV40. It does not encode large T antigen.
No.
That means that it doesn't have the traditional SV40 pathway for tumorigenesis. Sorry to all of those that have gone down that rabbit hole.
You hear people saying the SV40 virus is inside the... No, not the case at all.
It's just these little fragments, but it's enough. Having as we mentioned before, having an enhancer, which is a switch that acts at a distance to turn on other genes, a foreign enhancer, very powerful, one of the most powerful foreign enhancers known to molecular biology, that of SV40 that drives the large T antigen, is not a small thing. So it turns out that why Pfizer used a mammalian shuttle vector employing SV40 sequences makes absolutely no sense at all. You don't need that to produce RNA. If you were going to engineer, if you sat down tomorrow and said, hey, I'm going to build something that's going to allow me to make boatloads of mRNA, what are you going to do? And I want it to be stable. I want it to be, I can grow pounds of this stuff. And I want it to be stable. Well, how do you make a stable plasmid, which is what we're talking about, circular DNA, to produce the RNA as the template? How do you make it stable? Well, you make it as small as possible because you don't want a bunch of extra garbage in there that can yield recombination events that will make that more efficient for replication because bacteria will cut out any junk that they don't need to do the thing.
101, plasmid 101.
Yep. And so Pfizer didn't do that. They appear to have taken an off-the-shelf, 30-year-old technology. This is a shuttle vector that is exactly akin to the shuttle vectors that existed in the 80s. And use that to incorporate the SARS-CoV-2 engineered spike protein coding sequence. And there was no reason to have that extra garbage in there, but they did. So that raises the question whether that was foreknowledge or intent, or was it incompetence? And it's really hard to distinguish those things.
Yeah.
So that's one form of contaminant that you have in there. And because it's so large, it absolutely will recombine and you won't necessarily end up with a homogeneous product. And then there is the nuances of transcription using the T7 RNA polymerase. And that is the fidelity of that is imperfect. So you now have a synthetic in vitro system. So it's in the test tube. It doesn't have all of the cofactors that even phage T7 would have to ensure fidelity in the T7 RNA polymerase, nor do you have all the cofactors that would be present in a mammalian cell to ensure fidelity of transcription and translation. And so one of the things that happens, you don't have the bacterial factors, you don't have the phage factors, you don't have the mammalian factors. And so what you end up with is a really sloppy process. And among the things that happens there is that when you're using this double-stranded template to produce a single-stranded molecule, you end up with, I think the technical term is D-dimers, you end up with RNA-DNA hybrids because you don't have the enzyme present that normally would resolve those dimers, which are potentially a contaminant in regular transcription, which is the fancy word for making RNA out of DNA. These hybrids between a single-stranded DNA at the replication fork. So the DNA has to unwind just to make available one of the strands to generate the RNA off of. So that RNA that's generated can hybridize and produce a dimer, an RNA, a heterologous dimer, an RNA-DNA dimer, which the DNase I is typically used to degrade all the DNA after you do all this stuff because you want to get rid of it. So you don't transfect it. And that doesn't work perfectly, and that's why you end up with contaminants. But it doesn't resolve. It does not have the capability to digest these DNA-RNA hybrids. And so that's probably also driving the presence of the high-level contamination of the DNA. It's intrinsic to the fact that they're doing this in vitro. And the truth is that back in the day when I wrote that PNAS paper, I probably had this kind of contamination also.
No doubt.
And the truth is, as I think about it, this is one of the things, is I would run those gels to show that I'd gotten rid of the DNA, and often they'd had some really faint bands that I kind of just waved my hand around and said, well, it's almost all RNA. But it's probably, I didn't know any of this stuff back then. So probably that initial paper, well, the RNA is the predominant component of what I was transfecting with those studies that showed I could get a linear dose response in, R values, et cetera, for shooting the line. Probably there was some DNA contamination and probably some DNA RNA hybrid contamination.
So
also.
since we're on the topic of DNA contamination, can you speak a little bit to the potential of integration? This is one of those things that is often talked about, hand waved, not to worry, not going to happen. What is it? Has it been shown? Is it possible? What kind of danger might it pose?
So folks have actually taken now these preparations in vitro, in situ, I should say, in cell culture. And we have more nomenclature. When we say in vitro, generally we're talking about in a test tube. We say in situ, generally we're talking about in cultured cells in the world of cell biology and molecular biology. And in vivo refers to in humans or mice or critters, living things. So there's our nomenclature. So in situ experiments involving delivery, administration of the actual vaccine preparations. There are data now showing that integration of the DNA contamination can occur. Now, back in the day, long ago, there was a PNAS paper from the laboratory of Rudy Yanish in which he used HepG2 cells. This is very early on in the mRNA COVID vaccine story. So Rudy Yanish asserted that you could get reverse transcriptase. Remember, we talked about that before. The enzyme that makes a retrovirus work that can take DNA and turn it into RNA, or RNA and turn it back into DNA in particular. That's reverse transcriptase. So the backwards RNA to DNA. So Rudy asserted that you could deliver these RNAs into HepG2 cells, which is here's another word, transformed. So a cancer-like liver cell preparation that has been passaged for decades. Mm-hmm. So it's a long way away from being a liver cancer and has acquired a lot of genetic mutations and adaptations for growing in cell culture. So HepG2 cells are notorious for expressing line elements, which are kind of like historic retrovirus-like elements that encode something akin to reverse transcriptase. So Rudy did some experiments, and he asserted that you could put these RNAs onto HepG2 cells and get reverse transcription and integration. And a lot of people latched onto that. Problem with that is that Rudy has a long history, I'm sorry to say, of artifactual discoveries having to do with integration. So, and furthermore, using HepG2 cells is a long way away from using anything close to a living mammal. So that was the story until fairly recently, when after Kevin did his work and showed this contamination, the people went back and started to say, well, we don't have to look for reverse transcription. We can just look for whether or not these DNA fragment contaminants might be getting into the nucleus and into the chromosomes. And there are studies now showing that that's the case. I don't know if any of them passed peer review yet. But as you know, there's a lot of ways to do this and detect this. It's kind of almost a first or second year graduate level study to ask whether or not this happens. Very straightforward.
Fish.
And a lot of different ways to skin that cat using PCR or others. So there are some that are asserting that they have detected integration in cancers that have arisen associated with patients that have received these products. So then that goes down to a whole nother rabbit hole of whether or not those cancers are really associated with the vaccine. Are they just serendipitous, spurious artifacts? Are they associated with the virus itself? And generally, the way that this is approached is through monoclonal antibody staining that is specific for spike, and then you also use monoclonals targeted with some sort of a biochemical marker reaction to or a fluorescent tag that are detecting the presence of the surface. The nucleocapsid. So you do a combination nucleocapsid plus spike, differentially labeled antibodies, and if in the tumor you're seeing all the cells expressing spike but not nucleocapsid, then the logical conclusion is that you've had some sort of an integration event, or you have a clonal expansion. This goes back to the retrovirus days that I talked about. So this is what one would do back in the day with retroviral vectors trying to detect oncogenes. Is you'd look for a tumor that had typically by southern blot, a clear signal of the majority of the cells having the same genetic rearrangement. And then you'd go and hunt for what the surrounding genes were. Because you could probe for the retrovirus and then see if the tumor gave a single band for that retrovirus, then you know that's a clonal integration event. And then you could start asking, you could move out from there and say, okay, what is in the region of that integration event that might be a tumor suppressor gene or a positively acting oncogene, under the assumption that the LTRs, the promoter enhancers of the retrovirus, were turning on these surrounding genes. So that's how that went. And so that's kind of the basic idea behind detecting monoclonality in a tumor that's expressing spike but not nucleocapsid. Well, that would say that it's probably the vaccine, not the virus, because if it was the virus, then it would be both.
That's right.
And so having identified those kinds of phenotypes, then you go hunting. Do you see any sign of DNA associated with this plasmid sequence that Kevin has popularized? And then infer that you've had because if you see that, you typically would have to have, and you can do the sensitivity if you're using PCR, you'd be able to say, well, at least 10% of the cells have to have this integration event or whatever, have to have picked up this DNA fragment. And so there are some that are asserting that they have those data now. Again, very small n's, very anecdotal, hard science to do.
Yes.
As and it's clinical research, because you got to get permission, autopsy or postmortem or a biopsy from a solid tumor, typically, in order to even ask these questions. So it's going to be a long way coming before we can really definitively answer the question of whether you get integration. But from first principles, as you know, there is nothing more wickedly recombinogenic than a short DNA fragment.
Mm-hmm. Absolutely.
It is the perfect storm. And one of the things that people don't track with, this is another one of the misunderstandings that many lay people have, is the belief that these RNAs are going into all of your cells.
Right.
The whole idea back in the day, we're talking 87, 88. I didn't tell you another part of the story of the origin of the RNA vaccine concept. The postdoc working next to me at the bench was a guy named Dan St. Louis. And he was asking questions about the use of retroviruses to confer a reporter gene or a effector gene into a mouse model. And what he found, he did a thing where he would make little implants of transfected cells and then put those into the mice. And he would find that they would produce protein for about 2 weeks, the recombinant protein, and then it would stop. The whole laboratory was perplexed because this is a gene expression laboratory. They were working on FOSS and June. And, everybody was all about, the other part of the lab was all about gene expression. So obviously, there was some gene expression silencing event that was going on that was inactivating the expression off of these retroviral vectors. Everybody agreed that was the case. Having been through 2 years of medical school at that point in time, I opened up my medical textbooks and scratched my head. And what came out of that was, one ear out the other. What came out of that was, oh, dummy, this is an immune response. 2-week timeline. Okay. And then suddenly the whole logic of exogenous gene therapy, of the Ted Friedman concept, went right down the toilet bowl.
Right.
Because your body doesn't know that it's a good gene. It only knows that it's a different gene, your immune system, and it will reject it. So if you put in the muscular dystrophy gene, and you have a really efficient way to deliver muscular dystrophy gene into all the cells of your diaphragm, that's what kills these kids. For instance, those kids' immune systems aren't going to say, oh, goody, we finally got the right gene. They're going to say, no, this is something different from what I've seen before. I'm going to reject it and I'm going to attack it. Cellular immune response. And suddenly the gene therapy paradigm went right down the toilet. And the only applications since then that have worked at all have been delivery into immunoprivileged compartments or with immunosuppression, pretty much.
Right. Makes sense.
So that was the aha when this all happened was, okay, Robert, you got this whole new idea of using RNA as a drug, as an alternative gene therapy, and it has the advantage that it only lasts for a few hours. The RNA degrades. So you can treat it more like a drug. And if the patient has an adverse event, you just stop administering the drug. What's not to like? And what are you going to use it for? Because now the whole idea about gene therapy is kind of on the skids. Oh, I know. You can use it to produce an immune response. You can use it as a vaccine. Well, the thing about a vaccine is that you don't need very much antigen. So the fact that it's relatively inefficient suddenly becomes okay.
Right.
It's the entry level application for using mRNA as a drug in an incredible, the cationic lipid system, for all that it is in vivo, is incredibly inefficient. You can spend, you can spend a stupid amount of hours at a cryomicrotome, cutting sections, trying to find a blue cell.
Right.
Or a luciferase expressing
cell. We're trying to generate stable colonies with transfection.
So, but in the body, the immune system amplifies that signal. So what people don't understand is they think that, all of my cells now have spike protein. No. Well, if they do, it's because it's binding in separately because it's in the blood. But that's an error in assumption. That the truth is these particles are insanely inefficient. We just got a lot of cells, and you're administering a heck of a lot of number of particles, and so you get some delivery,
right? And
then the immune system amplifies the signal. So detecting an integration event is going to be wicked hard. Detecting what cells are actually producing the spike protein is going to be, generally speaking, wicked hard. And this whole idea that we've got gobs and gobs and gobs of this stuff infecting all our cells is just a false. It's a easily refuted hypothesis, but we still have enough. And this was the stunning thing to me, was when I ran the numbers early on. I could not. There was some data early on about the levels of spike protein being produced, and when I ran the numbers in terms of whatever it was, picograms or femtograms per microliter or something was what was being measured of spike protein. When I calculated based on blood volume for a human, I was like, oh my God, this is producing a way beyond the level of any expectation I ever had for efficiency of a system. So credit to the Arcturus et al. University of British Columbia crew for a far more efficient system than anything that I ever imagined might be possible. But it's still incredibly inefficient. But if you take a highly potent, biologically active protein like spike, you can amplify the immune response to it, and you can also produce a variety of adverse events associated with it. And then you combine it with this pseudouridine, for which the Nobel Prize was given. And that's an important one to unpack too. So Carrico and Weissman, I knew Katie Carrico. She objects to my using this term. I could say I coached her or mentored her or whatever. I gave her ideas. She called me early on and asked for some advice about how to get involved in the RNA world. And I introduced her to some people, and I talked to her about some of the key issues, invited her to a conference. And then this is all a decade after I'd done that stuff at the Salk. And her and Drew Weissman observed that the fundamental problem associated with this was the inflammatory response associated with the lipid nanoplex. And they inferred, and correctly, that a foreign RNA itself evokes an inflammatory response. This gets back to your fundamental question, along the lines of, can this ever be made safe and effective? So at the time, the science of pseudouridine, it's still in its infancy. It gets back to how complicated the mRNA world is. But the science of pseudouridine was really just breaking through. Just a few papers at the time when and one of the observations was that if pseudouridine is in RNA, it seems to downregulate the inflammatory response associated with foreign RNA. And so they said, bingo. we're going to take out the U in the reaction, AUGC, and add pseudo-U, synthetic pseudouridine, and incorporate pseudouridine randomly all the way through the backbone. And then we're going to test that. And they found that they got higher signal, presumably using luciferase. And Katie tells the story about how overjoyed they were when they ran the first experiment and showed that they were getting more signal. And so that was the aha. And the patent filed was for use for cancer. It had nothing to do with vaccines, by the way. And the thesis was that the pseudouridine was impacting on the inflammatory response to the foreign RNA. And so this was going to be the enabling concept. And Katie basically went on a national tour promoting this.
I recall.
And so, yeah, she's got an interesting background that I won't go into. But so it turns out that pseudouridine does a lot more than just impacting on inflammatory responses. And it seems to be a longer-term impact on downregulating immune responses, which is kind of a yin and the yang thing as it relates to this whole world. But among other things, pseudouridine really impacts on RNA folding and on frame shifting, which is this process we were talking about. Let's see if I can get it back again. The ribosome is the machinery. The RNA is threading through it and getting translated. And I talked about the transfer RNAs that are attached to amino acids that come in and dock the RNA as it's moving through. They can kind of misdock, and that's called frame shift, and get polymerized into the protein. But then if they do that, then from then on, the RNA is misread. It's shifted its reading frame instead of the one that it was supposed to be based on the starting codon to something else. And so then after that frame shifting event happens, what it produces is gibberish, unpredictable, up until it hits some random stop codon and the particle disassembles. So you end up with, when you add pseudouridine, and you end up with these frame shifting changes in the protein product. And the cell actually does that intentionally. It modulates in real cell biology. Pseudouridine is specifically placed at certain places in an RNA molecule after transcription, making RNA from DNA. There's a post-transcriptional modification process that places pseudouridine at certain places in the RNA to change how it folds. That folding can affect how efficiently it's translated. It can affect how long it lasts, how it stays before it's degraded. It affects a bunch of different things, and we only understand part of it because it's very RNA and context specific. And it seems that during pseudouridine modification may change with different cellular conditions, stress conditions, etc. So pseudouridine modification is used to control RNA translation. It's actually used to control frame shifting. It's used to control RNA stability. And it's used to control folding, this superstructure that RNA forms. So we end up with a logic that was promoted, a patent that was held by UPenn, where Carrico and Weissman are, that has generated, I think, billions of dollars for UPenn, that UPenn sold off to a holding company and is used in, just as the University of British Columbia tech is used in all of these products for the lipid nanoplex. The pseudouridine is used for the BioNTech and Moderna products, BioNTech becoming Pfizer. There was another company that was capitalized by a gentleman that you may have heard of before named Elon Musk. that did not use the pseudouridine tech. It had to do with licensing agreements, and they didn't think they needed it. And I think that was called CureVac. And early on in the cycle of the race to develop an RNA vaccine for COVID, that firm took a more methodical approach to dose optimization. And they produced an RNA that did not incorporate pseudouridine. So very much like the approach that I had used, giving rise to a much shorter half-life RNA. We now know that these pseudouridine modified RNAs, and it's not even natural pseudouridine, it's a synthetic version that they use, confer very long-term stability to the RNA. Still don't know how long, but instead of hours, it's certainly weeks, if not months, maybe years. So this other company didn't do that, and they started at a lower dose in their initial clinical trials, in their phase 1 clinical trials. And Moderna and Pfizer used a much higher dose, Moderna being the highest of all in their initial clinical trials. And not surprisingly, the CureVac antibody titers were lower
makes sense.
than the other two. And so the conclusion was that the CureVac product didn't work as well because it didn't have pseudouridine, obviously. Well, actually, if you go back and look at the data and you adjust for dose, the antibody response is pretty similar. So the question comes up, do you really even need the pseudouridine, which is absolutely associated with these frame-shifting artifacts, the production of surreptitious proteins of unknown function that are probably inflammatory, the long half-life, et cetera, et cetera. And of course, this is the basis for the Nobel Prize. You were going to say something about that. And I'll give you a little story about that. So way back in the day, like in 91, I was invited to the laboratory of Dr. Peter Lillystrom at the Karolinska to talk about the work that I had done, because Peter was working together with his graduate student at the time, Peter Berglund, who then became my postdoc that I was mentioning earlier in the story. So Lillystrom invited me out to the Karolinska to talk about RNA delivery because he was working on, drumroll, rimshot, self-replicating RNA based on alpha viruses, which is, of course, the new tech that everybody is going to, as you can greatly reduce the dose. And what could possibly go wrong with a self-replicating RNA? That's tongue-in-cheek. So Peter invited me out, and I talked about what I had done. And we had maintained intermittent communication over the years. And about the time when I went on the Brett Weinstein podcast, there was this ramp up of PR, clearly designed to get the Nobel Prize for UPenn and Kariko and Weissman. And I, having been around the Nobel laureate world at the Salk, et cetera, I was very familiar with how these PR campaigns are run by people that want to get the prize. So I saw this playing out, and I called up Peter and I said, they're taking credit for my invention, and they're saying that Katie and Drew did this all by themselves. They were the original inventors. This, interviews on CNN and the New York Times, et cetera. I found this a little offensive. It's kind of stolen valor. And I said, Peter, they're going to, should I try to spin up some sort of a press campaign here? And he happened to be on the Nobel Prize Committee at that time. And he said, Robert, don't worry about it. They've already reviewed, we're aware of this. They've been nominated. We've already reviewed that nomination, and their contributions don't rise to the level of meriting the award. And I've made everybody aware of what your role was in all this. And in fact, that year, the award went to somebody completely different. And then the next year, a new committee came in that I've been told is very woke. There was major investments in the region around the Karolinska in RNA vaccine commercial development and donations to the Karolinska. And that year, Grieco and Weissman were awarded not for the technology, but for their role in enabling these vaccines. And the committee said that the reason why was because the reason they justified the award was they wanted to encourage more people worldwide to accept the product, and getting the basically seal of approval of the Nobel Prize Committee would encourage more people to accept the vaccine product. So they awarded it for the vaccine product and the role of Grieco and Weissman, which actually they didn't play much of any role in the development of the actual vaccines. As we now know, for instance, the Moderna product was engineered at the NIAID at the Vaccine Research Center. And a lot of this was actually done through subcontractors. But that's what happened. They're not going to give the Nobel Prize twice to the same topic. And you move on. And in many ways, it's if I had been the third wheel on that prize, then I would be dealing with the Robert. I would have even more people accusing me of being a mass murderer than already do.
Right.
And, yeah. So
well said.
It is what it is, and I make my living now from Substack and trying to influence policy, and I'm grateful for the occasional opportunity to talk about the good old days like you're doing here.
It's absolutely my
and
pleasure.
Technology, but most people want to talk about the World Economic Forum and the Great Reset and the fact that there's no virus at all.
Right.
And all these other interesting theories.